The effect of temperature on enzyme activity. Juliana, Colin, Armin, Jimmy. Introduction. Enzymes are biological catalysts that speed up chemical reaction in living organisms. Each enzyme has an optimal temperature range in which it functions most effectively. Temperature changes can affect the rate of enzyme activity, potentially leading to denature at extreme temperatures. Understanding how temperature affects enzyme activity can provide insight into metabolic processes and the functioning of organisms under varying environment conditions. Hypothesis and prediction. Hypothesis. Temperature affects enzymes activity because enzymes have an optimal temperature range for maximal activity. Prediction. If the temperature increases, then the enzyme activity will increase up to optimal point, after which it will decrease due to denature.
Methods. Here's how we tested the temperature effects of enzyme activity. First, we prepared the enzyme and substrate solutions with constant concentrations. Then we set up reaction mixtures and divided them into groups, incubating each at different temperatures: 10 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, 50 degrees Celsius, and 70 degrees Celsius. After incubation, we measured enzyme activity using a spectrophotometer to see how much product was formed. Finally, we compared the activity at different temperatures to find the optimal one.
So these are our results. In table one, this is just a table of our data. You could see as temperature goes up, so does enzyme activity until 37 degrees is reached, and then it starts to go back down. And then in figure one is the same thing. It's just we just made it into a graph. As temperature goes up, so does enzyme activity until 37 degrees is reached, and then it starts to go down.
The result text is the enzyme activity increased, as we see, from 10 degrees Celsius to 37 degrees Celsius, with the highest activity being observed at 37 degrees Celsius. Beyond 37 degrees Celsius, the enzyme activity decreased with a significant drop at 70 degrees Celsius, indicating denaturation of the enzyme at high temperatures. Compared to the control group at 25 degrees Celsius, the enzyme activity was higher at 37 degrees Celsius and lower at both 10 degrees Celsius and 70 degrees Celsius. Our conclusion, as stated in the introduction, enzymes are a biological catalyst with optimal temperature ranges for maximal activity. The experiment aimed to investigate how temperature affects enzyme activities, hypothesizing that activity would increase with the temperature up to an optimal point and then decrease due to denaturation. The results supported our hypothesis, showing that the enzyme activity increased from 10 degrees Celsius, peaked at 37 degrees Celsius, and decreased above 37 degrees Celsius, indicating denaturation. This aligns with the understanding that enzymes have an optimal temperature range for activity. The control group at 25 degrees Celsius showed lower activity than at 37 degrees Celsius, confirming the optimal temperature peak. The limitations included potential inaccuracies in temperature maintenance and measurement errors, assuming constant enzyme and substrate concentrations. Understanding enzyme temperature dependence has important implications, such as optimizing industrial enzyme processes and understanding metabolic adaptations in organisms. Further research could explore other factors like pH and substrate concentrations to provide a more comprehensive understanding of enzyme kinetics.
Limitations and assumptions. Several limitations and assumptions could have affected the results. Inaccuracies in maintaining the exact temperature throughout the experiment could have introduced variability in the data. Measurement errors such as incorrect pipe tuning or spectrophotometer calibration could have also influenced the results. it is assumed that the enzyme and substrate concentrations remain constant throughout the experiment.
Perfect. So that was our presentation, and that's it. Thank you all.